Hello, you're all very welcome. So today I'm going to go over the practical assignment for Immunology NU2 301. So what I've provided in the PowerPoint, which is available on Blackboard Learn, are 4 links to documents that you'll need to complete this assignment. So the first link is a link to a document related to the background information for the practical. Okay, folks, so for this practical, you have to do a little bit of reading to update your knowledge on HIV and also cell counting and the technical aspects of flow cytometry. Another link which I provided is a link to a document related to laboratory instructions. Now, unfortunately, due to COVID, of course, we can't undertake this practical on campus. So the laboratory instructions are there for your own reading and to give you an indication of the type of technical aspects of the practical that would have taken place. But you will need the laboratory instructions for specific details related to your calculations. So please note this. Another thing which I want to emphasize now and I'll emphasize later is be wary of dilution factors and the need to adjust your calculations for dilution factors, okay? And to work out the dilution factors, you'll need the information in the laboratory instructions. The third document which I'll be providing you with a link to is a presentation on the theory behind flow cytometry and HIV, okay? So that link is around an hour long and will enable you to get some level of understanding of flow cytometry, and it will help you answer some of the technical questions in the answer booklet. Of course, lastly, the link which will be provided last is the answer booklet itself. Okay, folks, so the answer booklet's there. Complete it, submit it to Blackboard. And another thing which I want to emphasize now and I'll emphasize later is be wary of plagiarism. Okay, folks, if you plagiarize, it will be detected. You will have marks deducted, and you will then have to go through the disciplinary route associated with plagiarism. So please try to ensure that you don't plagiarize in this assignment. Okay, in relation to the actual practical, there are 2 tasks involved in assignment 2. Task 1 involves peripheral blood mononuclear cell isolation from blood and a subsequent cell count. And task 2 involves flow cytometry analysis in HIV patients, which we'll cover later in the lecture. So, in relation to task 1, this hypothetical scenario is that Emer would like to investigate the effects of the anti-inflammatory activity of N3 polyunsaturated fatty acid, DHA. So what Emer wants to do is isolate the peripheral blood mononuclear cells, which essentially are the monocytes and lymphocytes, from human blood. And what she wants to then do is, once they're isolated, she will culture these cells with and without DHA for 24 hours. And when she's finished that, she will measure the amount of IL-6 that is being released by these cells. And the ultimate aim would be to determine whether DHA can reduce the amount of pro-inflammatory IL-6 produced. Okay, so of course this is all hypothetical. So she's going to incubate the isolated peripheral blood mononuclear cells in 100 micromolar of DHA. Okay, so this is what has happened on the day. Emer has isolated the peripheral blood mononuclear cells from the blood and wants you to help her set up the experiment. So she's provided you with 5 mL of solution containing peripheral blood mononuclear cells in the cell buffer. And what she's asking you to do as a student is to determine the number of peripheral blood mononuclear cells per mil in the solution that she's provided you with. And she wants you to do this by carrying out a cell count using a hemocytometer. Once you've done the cell count, Emer would like you to then subsequently give her 2 mils of cell buffer containing 5 by 10 to the 5 peripheral blood mononuclear cells per mil. Okay, now in terms of peripheral blood mononuclear cells, I'm going to show you a short video as to how Emer managed to isolate these peripheral blood mononuclear cells.
The following video is a technical guide that demonstrates how to isolate mononuclear cells from whole blood by centrifugation through a density gradient medium such as Ficolpac Plus. To begin, ensure all reagents are at room temperature. Dilute the blood sample at a 1:1 volume ratio with the appropriate medium. Add a volume of density gradient medium to a fresh tube according to the specifications for that density gradient medium. Gently layer the diluted blood on top of the density gradient medium. Take care not to mix the two layers. Centrifuge for 30 minutes at 400 G with the brake off. Carefully harvest the cells by inserting the pipette directly through the upper plasma layer to the mononuclear cells at the interface. Alternatively, you can first remove the upper layer and then collect the cells. Finally, wash the harvested cells twice in the appropriate buffer. The cells are now ready for easy-step cell separations or other downstream applications. For more information on blood sample processing and cell isolation, please visit stemcell.com.
So in terms of the procedure to isolate the peripheral blood mononuclear cells, it involves taking 5 mils of blood and mixing it with an equal amount of PBS, phosphate buffered saline. You then layer it on the Fickle density media, and the Fickle density media basically lets you separate the blood based on the mass of the different components. Okay, and we'll talk a little bit more about that in a minute. So once you've layered the blood on the Fickle density media, you centrifuge it at 400 G. And once the centrifugation has completed, you then isolate the peripheral blood mononuclear cell layer. And once you've isolated the peripheral blood mononuclear cell layer, you mix it with PBS to wash it. You then centrifuge it at 300 G, and what you'll have is at the bottom of the centrifuge tube, you'll get a cell pellet. That is, the cells have been stuck to the bottom of the tube, and you remove the supernatant, that is the PBS, and now you have a pellet of cells at the bottom of your tube, and you resuspend the pellet of cells in 5 mils of PBS, and that is the solution that Emer has provided you with. A 5 mil PBS solution containing the peripheral blood mononuclear cells that has been isolated from the blood. So the only thing that you have to do now is count how many cells are present in this tube. So this is a visualization of the tubes that Emer would have used to isolate the blood mononuclear cell. So if you look at the first tube to the left, you can see that the blood and PBS has been layered on the Fickle separation gradient media. Okay, so the blood essentially just sits directly on top or floats on top of the Fickle media. Then after the centrifugation, as you can see the tube in the middle, after centrifugation, all the components separate out based on mass. So at the top you have the yellow plasma, and just at the bottom of the plasma level, you'll have the peripheral blood mononuclear cell layer. And this layer kind of floats as a kind of viscous layer below the plasma layer. Below the peripheral blood mononuclear cell layer, you then have the Fickle gradient media that separates the peripheral blood mononuclear cell from the next sedimented level, which contains erythrocytes and granulocytes. Okay, and that is the after the centrifugation step. So what Emer then subsequently did was remove the peripheral blood mononuclear cell and put it into a fresh tube. Okay, and she then took that tube with the peripheral blood mononuclear cells and the PBS, and she centrifuged it to pellet the cells at the bottom of the tube. Then she removed the supernatant, the PBS, and she was left with the peripheral blood mononuclear cells adhered to the bottom, and she subsequently added 5 mL of PBS to resuspend the cells in solution. Okay, so now that we have the peripheral blood mononuclear cells in PBS in this tube, we now want to undertake a cell count. And the way we undertake a cell count is by taking 50 microliters of the cell solution, putting it into an Eppendorf, and then subsequently taking 50 microliters of the cell dye trypan blue and putting it into the Eppendorf, and subsequently mixing up and down to ensure that we get an equally dispersed solution. And what we then do is we take 10 microliters of the liquid from the Eppendorf, and we add it to the hemocytometer. Okay. And when we add it to the hemocytometer, we're doing it to ensure that we are able to undertake a cell count. Now, a hemocytometer is a device that enables us to quantify the amount of cells present per microliter, sorry. And what a hemocytometer basically is, it's like a little reservoir that enables you to put in a defined volume of liquid and subsequently calculate the amount of cells per volume of liquid. Now, when you get a hemocytometer, what you'll notice is most hemocytometers that you will work with will have 2 chambers. It'll have a top chamber, as you can see here, and it'll have a bottom chamber, which you can see here. Now, the 2 chambers are the exact same, and they're there to basically enable you to do 2 cell counts at the same with the same sample for reproducibility and quality control measures. Okay, as I said, the hemocytometer is a chamber that allows you to add a defined volume of liquid. Now, in terms of the counting chambers, this is what you would see if you look down a microscope. You'll see these, if you see here, you'll see that a whole range of different types of squares. Now, in terms of the cell count that you're undertaking, you want to focus on these squares here. Okay. Now, in order to obtain a cell count, what you have to do is take the average number of cells present in this part of the grid, an average count in this part of the grid, this part of the grid, this part of the grid, and this part of the grid. Once you take your cell count, you take the average, and when you get your average count, what you have is the average count per mil. And the way you calculate this, it's the average count by 10 to the 4. So if you get an average count of 35, then you have a cell count of 35 by 10 to the 4 cells per mil. Now, I said at the start of the discussion, one thing we have to account for is dilution factor. Now, the fact that we diluted the cells in trypan blue in a 1-to-1 ratio basically means that we have to multiply the cell count that we got by the dilution factor to get the true number of cells per mil. Okay, so always ensure that whatever cell count you get, you multiply by the dilution factor, and then you subsequently have a true cell count, i.e. the number of cells that are present in the solution, in this case that was given to you by Emer. So what would it look like? This is what the grid that you'll be provided with on the day of the practical will look like. Okay, so as you can see, it's the same as before. Sorry, it's 4x4 grids, 4x4, to identify live cells and dead cells. Okay, so how do we determine if a cell is live or not? So the concept of the trypan blue viability test is that cells with an intact membrane will not allow the trypan blue to enter the cells. Okay, whereas dead cells that have disrupted cell membrane will take up the dye, and subsequently you'll be able to tell the difference between a live cell and a dead cell. So how do you visually identify a live cell compared to a dead cell? A live cell will have a very clear defined membrane, almost like a halo on the outside of the cell, as you can see here. Whereas a dead cell will be colored with dye and will have a faded membrane. Okay, and that is how you will define live and dead cells. So if we look at this first square, I can see that we have 1, 2, 3, 4, 5 viable cells. Okay, you see the little halo around the cell. Whereas in this square, we have 1, 2 cells that are not viable. How do we tell they're not viable? Because they don't have a defined membrane, and you can see there's a difference in the staining of. Okay, at this point, we're going to proceed to watch a video which will give us an overview of how to use a hemocytometer to determine a cell count, and also how to determine cell viability, both of which will be useful for the practical write-up. Okay.
In this video, you will see how to use a hemocytometer to get an accurate cell count and how to convert this data into usable cell density and viability information. The hemocytometer was originally developed for counting blood cells. Today, it is used to count many other cell types as well. A hemocytometer consists of a special glass microscope slide that contains 2 separate counting chambers. Each chamber is etched with a carefully crafted grid with exact dimensions. There are 2 supports on either side of the counting chamber. When a cover glass is placed on the supports, the depth of the chamber will be exactly 0.1 millimeters. Now we know both the area and the depth of the counting chamber, and we can calculate the volume. We can also use the grid to count the number of cells in each section and calculate the cell density. Before you start, you need to dilute your cell suspension in trypan blue solution, which is used to stain any dead cells present in your sample. Make sure your sample is mixed well. Counts should be within 2 minutes of incubation with trypan blue. Be sure not to wait more than 5 minutes, as longer incubations will result in trypan blue entering live cells. A dilution factor between 2 and 10 usually works well for mammalian cells. To get accurate cell counts, it is recommended to have no fewer than 10 and no more than 50 cells over each square millimeter. Basically, you don't want to be counting 100 or 200 cells per square. You want to use appropriate dilutions to get 10 to 50 cells per square. Make sure the hemocytometer and cover slip is clean. 70% ethanol can be used for the cleaning. Carefully place the cover slip on the supports using gentle pressure and small circular motions. Here's a tip: moisten the shoulders of the hemocytometer before you fix the cover slip. Make sure your diluted cell sample is mixed well and load 10 microliters by gently resting the end of the pipette tip at the edge of the chamber or the edge of the cover slip. Do not overfill the chamber. Let the liquid be drawn out of the tip and fill the counting chamber by capillary action. With a microscope and a 10 times magnification, you will be able to see approximately 1 square per viewing window. Use phase contrast to distinguish the cells. Live cells will not be stained by trypan blue and will appear white. while any dead cells will be faint or dark blue. Now you can count all the cells that fall within a couple of the larger squares and include any cells on the right hand or bottom boundary line. Count the number of live cells and dead cells. A good suggestion could be to count the cells in squares 1, 3, 5, 7 and 9. Obviously, the more squares you count, the more accurate your cell count will be. Just remember how many squares you've counted. Did you know there's an app you can use for this? The Cell Culture app has a cell counter tool that will help you keep track of based on the number of viable and dead cells. You can now calculate the cell viability. To calculate the density of the viable cells, you need to determine the number of live cells in a particular volume. This volume is defined by the number of squares that you counted and the volume of one such square. We also multiply by the dilution factor in order to determine the cell density of your original cell solution prior to diluting with trypan blue solution. The hemocytometer is designed so that the volume of each of the largest squares is constant. Each square will hold a number of cells equivalent to the number of cells counted multiplied by 10 to the power 4 per milliliter. To convert, we then calculate the density of viable cells by looking at the number of live cells multiplied by 10 to the power 4 per milliliter, the number of squares counted, and the dilution factor. The hemocytometer and these equations help us determine cell density and viability. Based on this, you can also create growth curves or calculate the best window for cell passaging. You'll find more information on how to perform these basic cell culture calculations in a separate video.
So now we're going to deal with the issue of calculating the volume of cells that is needed in order to fulfill the request that Emer has asked for. So, for instance, if Emer had asked to provide 2 mils of 5 by 10 to the 5 cells per mil, you need to work out the total number of cells that are required. Okay. Say, for instance, the cell count you obtained was 45 by 10 to the 4. Then in order to work out how many cells we need to achieve 2 mils of cells at 5 by 10 to the 5, we need to follow the 3 steps that we're about to cover. Okay, so step 1 is, what is the total number of cells required? Okay, and the equation for that is volume of cells required, multiplied by cell concentration desired. Okay, in this case, we've been asked to provide 2 mils at 5 by 10 to the 5 cells per mil. And when you do that calculation, it tells us that we need 10 by 10 to the 5 cells in total, or another way of expressing it would be 1 by 10 to the 6. Okay, therefore, in order to obtain 2 mils at 5 by 10 to the 5, we're going to need 1 by 10 to the 6 cells. Okay, so we now know the number of cells required for that. Step 2, how much of my cell solution do I need to obtain 1 by 10 to the 6 cells? So at this step, you're trying to work out how much of the cell solution obtained from Emer do you need to obtain 1 by 10 to the 6 cells. And the way to do this is cell number required divided by cell count obtained. So the cell number required is 1 by 10 to the 6, and the cell count obtained was 45 by 10 to the 4, or 4.5 by 10 to the 5, whatever way you want to express it. Okay, but the easiest way to do this calculation is to make sure that the cell number required and the cell count obtained are in the same units. Okay, so I've transformed both of these to 10 to the 4. So it's 100 by 10 to the 4 divided by 45 by 10 to the 4. So it's 100 divided by 45, which means the answer is we need 2.222 mils of cells that we obtained from Emer to obtain 1 by 10 to the 6 cells in total. Okay. Step 3. Remove the 2.222 mils of cells, place them into a centrifuge tube, centrifuge them, remove the supernatant, and when you remove the supernatant, at the bottom of the tube you'll have the pelleted cells. And with the pelleted cells at the bottom of the tube, simply add 2 mL of buffer to resuspend the cells. And what you then have is 2 mL of buffer containing 1 by 10 to the 6 cells in total. And because it's in 2 mL, the concentration then is 5 by 10 to the 5. So that's how you go about doing a cell count and working out the volume of cells that you need to achieve a given concentration. Okay. So in terms of what you're being asked to do in relation to task 1 for assignment 2, task 1 of the assignment task 1 of the assignment will provide you with a hemocytometer screenshot, and we're asking you to use that screenshot to undertake a cell count. Okay, so you undertake a cell count using the screenshot, and what I'm asking you to do is to determine the percentage cell viability. I'll also ask you to calculate the volume of cell solution provided by EMER that will be required to obtain enough cells for an experiment. Okay. For example, you will be asked to determine the volume of the cell solution provided by EMER that would be required to obtain a solution of 2 mL at 10 by 10 to the 5 cells per ml. Okay, and that part of the assignment will be worth 20 marks. Okay, and you'll find this in the answer book. So that's task 1. For task 2, it will be focused on flow cytometry. You'll be provided with blood from 3 HIV patients, and you'll be asked to quantify the absolute number of CD4 cells and CD8 cells, and from this provide an indication of HIV severity. What's involved is the clinician would obtain a blood sample from the patient. They would then add an antibody mix to the blood to enable them to identify a range of cell markers that would allow them to identify CD4 and CD8 cells. The antibody labeled blood will then subsequently be analyzed using a flow cytometer, and that will enable us to quantify the absolute number of CD4 and CD8 cells per microliter of blood. And you'll also be asked to calculate the ratio of CD4 to CD8, because this is also important in the clinical decision. Now, there's a lot more information on the background to the flow cytometer in the presentation provided on BBLearn and on the link on this presentation. Okay, in terms of the procedure, the clinician would take a specialized tube called a true count tube, which contains in it counting beads, which we'll cover shortly. So they would take this true count tube and they would add 50 microliters of antibody. They would then subsequently add 50 microliters of patient's heparinized blood sampled to a true count tube. And after that, they would add 900 microliters of BD lysine solution to a true count tube. They would subsequently vortex the mixture, let it incubate in the dark for 5 minutes, and after that, the blood sample is ready to be analysed on the flow cytometry machine. In terms of determining the volume of blood used, this is an important factor we need to consider because determining the volume of blood that's been used by the flow cytometer is critical to establishing the amount of CD4 and CD8 cells per volume of blood, and to enable us to compare to guidelines that are provided on this. So, flow cytometer itself takes up blood for analysis, but the difficulty is that when the flow cytometer takes up the blood for the analysis, there's no way of determining what volume was taken up by the machine. So this creates a problem because the clinical decision needs to be based on how many CD4, CD8 cells there are per microliter of blood. So, as I said, we need to determine the volume of blood used by the flow cytometer. Well, how do we do that? We're able to quantify the volume of blood used by the machine by spiking the blood with a known number of true count beads. Okay? These true count beads have fluorescent properties that enable the flow cytometer to detect them. So, for instance, if we spike 1 ml of blood with 1 million beads, okay, so the patient blood now contains 1 million beads. If the flow cytometer detects 500,000 beads when the sample is analysed, then we can establish what volume was used by the flow cytometer. In basic terms, if 1 ml of blood contained 1 million beads and the flow cytometer detected 500,000, then on that basis we can allude to the fact that most likely 0.5 mils of the blood sample was used by the flow cytometer. And that's the basic principle for the true count beads. They're fluorescent beads that are spiked in the blood, and the flow cytometer then detects the amount of these fluorescent bloods that's taken into the machine, and indirectly then we can work out the volume of blood that was used by the flow cytometer. So what I've tried to do here then is provide you the step-by-step procedure for calculating the actual number of CD4 and CD8 cells present in the blood to enable you to make clinical decisions. Okay, so the calculation, you need to calculate the volume of liquid or blood taken from the true count tubes by the flow cytometer in order to determine an absolute number of CD4 and CD8 cells. And as I said before, this is achieved by true count beads. Now folks, as I noted earlier at the start of the presentation, in order to get the true number of cells in blood, you need to take account of dilution factors. Okay, so the blood was mixed with antibody and mixed with lysine solution. So you need to take that into account at the final calculation, okay, for the dilution factor. So, in order to work out how many beads per microliter or how many bead counts per microliter of bead solution in tube, it's a total number of beads divided by the final volume, okay? And then the volume used by the machine, the calculation for that is the bead count that the machine has detected divided by the bead count per microliter, okay, which you've calculated just above. Okay, so that will give you an indication of the volume used. So now, in terms of absolute count, you will be provided with results and you'll be asked to subsequently determine the absolute count. And the absolute count will be the gated cell count on the flow cytometer, okay, the number the number of cells that the results report, divided by the volume used, okay. And then, as I said before, once you have that absolute count, you need to adjust for the dilution factor. So the absolute count multiplied by the initial dilution factor will let us know the actual number of cells per microliter of blood. And of course, as I said previously, another important factor to consider is the CD4 to CD8 ratio. Okay, so a CD4 to CD8 ratio will enable us to determine how sick these individuals are. And there's a worked example in the notes provided that have been provided alongside this presentation that will enable you to undertake these calculations. Okay, folks, so that's just a basic introduction in relation to what is expected from you in relation to this assignment or practical. So in terms of tips and advice, take the time to view the presentation on the theory of the flow cytometer, and also read the associated documents to help you with this assignment. It's an assignment, therefore you have to invest time to do some background reading. At every stage that involves calculation, always remember to consider dilution factors. And like any calculation, it's important to show all you're working out, each step of your working out, in order for you to achieve maximum marks. Another tip is be careful with plagiarism. If cases of plagiarism will be detected, they've been detected before and will be detected again. And if plagiarism is detected, then you will be required to undertake a plagiarism interview, and any component that was plagiarized will not be marked. So if you plagiarize, you will not receive a mark, and you'll have to resit with a cap at 40%. So take care with plagiarism. And lastly, if there's any clarity needed on any aspect of the presentation or the practical, please don't hesitate to contact me. I'm more than happy to have a Blackboard Learn Collaborate Ultra discussion if that's needed in order to provide some clarity on that. Aside of that, folks, it's not the most ideal way to get introduced, but I really look forward to interacting with you in relation to this module and this assignment. Thank you very much.